In 19th century America, practically everything that was built involved wood. Pine was especially attractive for building purposes. It is durable and strong, yet soft enough to be easily worked with even the simplest of hand tools. It also floats nicely on water, which allowed it to be transported to distant markets across the nation. The central and northern reaches of the Great Lakes states, Michigan, Wisconsin, and Minnesota, all contained extensive pine forests as well as many large rivers for floating logs into the Great Lakes, from where they were transported nationwide. By 1860, the settlement of the American West along with timber shortages in the East converged with ever-widening impact on the pine forests of the Great Lakes states. Over the next 30 years, lumbering became a full-fledged enterprise in Michigan, Wisconsin, and Minnesota. Newly formed lumbering corporations bought up huge tracts of pineland and set about systematically cutting the trees. Both the colonists and the later industrialists saw timber as a commodity, but the latter group adopted a far more thorough and calculating approach to removing trees. In this sense, what happened between 1860 and 1890 represented a significant break with the past. No longer were farmers in search of extra income the main source for shingles, firewood, and other wood products. By the 1870s, farmers and city dwellers alike purchased forest products from large manufacturing companies located in the Great Lakes states rather than chopping wood themselves or buying it locally. The commercialization of lumbering was in part the product of technological change. The early, thick saw blades tended to waste a large quantity of wood, with perhaps as much as a third of the log left behind on the floor as sawdust or scrap. In the 1870s, however, the British invented bandsaw, with its thinner blade, became standard issue in the Great Lakes states lumber. Factories, meanwhile, the rise of steam-powered mills streamlined production by allowing for the more efficient, centralized, and continuous cutting of lumber. Steam helped to automate a variety of tasks, from cutting to the carrying away of waste. Mills also employed steam to heat log ponds, preventing them from freezing and making possible year-round lumber production. For industrial lumbering to succeed, a way had to be found to neutralize the effects of the seasons on production. Traditionally, cutting took place in the winter, when snow and ice made it easier to drag logs on sleds or sleighs to the banks of streams. Once the streams and lakes thawed, workers rafted the logs to mills, where they were cut into lumber in the summer. If nature did not cooperate, if the winter proved dry and warm, if the spring thaw was delayed, production would suffer. To counter the effects of climate on lumber production, loggers experimented with a variety of techniques for transporting trees out of the woods. In the 1870s, loggers in the Great Lakes states began sprinkling water on sleigh roads, giving them an artificial ice coating to facilitate travel. The ice reduced the friction and allowed workers to move larger and heavier loads. But all the sprinkling in the world would not save a logger from the threat of a warm winter. Without snow, the sleigh roads turned to mud. In the 1870s, a set of snowless winters left lumber companies to ponder ways of liberating themselves from the seasons. Railroads were one possibility. At first, the remoteness of the pine forests discouraged common carriers from laying track, but increasing lumber prices in the late 1870s combined with periodic warm, dry winters compelled loggers to turn to iron rails. By 1887, 89 logging railroads crisscrossed Michigan, transforming logging from a winter activity into a year-round one. Once the logs arrived at a river, the trip downstream to a mill could be a long and tortuous one. Log jams were common, at times stretching for 10 miles, and became even more frequent as pressure on the northern Midwest pinelands increased in the 1860s. To help keep the logs moving efficiently, barriers called booms were constructed to control the direction of the timber. By the 1870s, lumber companies existed in all the major logging areas of the northern Midwest. 46-2 The commercial revolution in medieval Europe beginning in the 1160s, the opening of new silver mines in northern Europe led to the minting and circulation of vast quantities of silver coins. The widespread use of cash greatly increased the volume of international trade. Business procedures changed radically. The individual traveling merchant who alone handled virtually all aspects of exchange evolved into an operation involving three separate types of merchants: the sedentary merchant who ran the home office financing and organizing the firm's entire export-import trade, the carriers who transported goods by land and sea, and the company agents resident in cities abroad. who, on the advice of the Home Office, looked after sales and procurements. Commercial correspondence, unnecessary when one businessperson oversaw everything and made direct bargains with buyers and sellers, multiplied. Regular courier service among commercial cities began. Commercial accounting became more complex when firms had to deal with shareholders, manufacturers, customers, branch offices, employees, and competing firms. Tolls on roads became high enough to finance what has been called a road revolution, involving new surfaces and bridges, new passes through the Alps, and new inns and hospices for travelers. The growth of mutual trust among merchants facilitated the growth of sales on credit and led to new developments in finance, such as the bill of exchange, a device that made the long, slow, and very dangerous shipment of coins unnecessary. The ventures of the German Hanseatic League illustrate these advancements. The Hanseatic League was a mercantile association of European towns dating from 1159. The league grew by the end of the 14th century to include about 200 cities from Holland to Poland. Across regular, well-defined trade routes along the Baltic and North Seas, the ships of League cities carried furs, wax, copper, fish, grain, timber, and wine. These goods were exchanged for finished products, mainly cloth and salt, from western cities. At cities such as Bruges and London, Hanseatic merchants secured special trading concessions, exempting them from all tolls and allowing them to trade at local fairs. Hanseatic merchants established foreign trading centers, the most famous of which was the London Steelyard, a walled community with warehouses, offices, a church, and residential quarters for company representatives. By the late 13th century, Hanseatic merchants had developed an important business technique, the business register. Merchants publicly recorded their debts and contracts and received a league guarantee for them. This device proved a decisive factor in the later development of credit and commerce in Northern Europe. These developments added up to what one modern scholar has called a commercial revolution. In the long run, the commercial revolution of the High Middle Ages brought about radical change in European society. One remarkable aspect of this change was that the commercial classes constituted a small part of the total population, never more than 10%. They exercised an influence far in excess of their numbers. The commercial revolution created a great deal of new wealth, which meant a higher standard of living. The existence of wealth did not escape the attention of kings and other rulers. Wealth could be taxed, and through taxation, kings could create strong and centralized states. In the years to come, alliances with the middle classes were to enable kings to weaken aristocratic interests and build the states that came to be called modern. The commercial revolution also provided the opportunity for thousands of agricultural workers to improve their social position. The slow but steady transformation of European society from almost completely rural and isolated to relatively more urban constituted the greatest effect of the commercial revolution that began in the 11th century. Even so, merchants and business people did not run medieval communities, except in central and northern Italy and in the county of Flanders. Most towns remained small. The nobility and churchmen determined the predominant social attitudes, values, and patterns of thought and behavior. The commercial changes of the 11th through 14th centuries did, however, lay the economic foundation for the development of urban life and culture. 41-3 Trade and early state formation. Bartering was a basic trade mechanism for many thousands of years, often sporadic and usually based on notions of reciprocity. It involved the mutual exchange of commodities or objects between individuals or groups. Redistribution of these goods through society lay in the hands of chiefs, religious leaders, or kin groups. Such redistribution was a basic element in chiefdoms. The change from redistribution to formal trade, often based on regulated commerce that perhaps involved fixed prices and even currency, was closely tied to growing political and social complexity and hence to the development of the state in the ancient world. In the 1970s, a number of archaeologists gave trade a primary role in the rise of ancient states. British archaeologist Colin Renfrew attributed the dramatic flowering of the Minoan civilization on Crete and through the Aegean to intensified trading contacts and to the impact of olive and vine cultivation on local communities. As agricultural economies became more diversified and local food supplies could be purchased both locally and over longer distances, a far-reaching economic interdependence resulted. Eventually, this led to redistribution systems for luxuries and basic commodities, systems that were organized and controlled by Minoan rulers from their palaces. As time went on, the self-sufficiency of communities was replaced by mutual dependence. Interest in long distance trade brought about some cultural homogeneity from trade and gift exchange, and perhaps even led to piracy. Thus, intensified trade and interaction, and the flowering of specialist crafts, in a complex process of positive feedback, led to much more complex societies based on palaces, which were the economic hubs of a new Minoan civilization. Renfrew's model made some assumptions that are now discounted. For example, he argued that the introduction of domesticated vines and olives allowed a substantial expansion of land under cultivation and helped to power the emergence of complex society. Many archaeologists and paleobotanists now question this view, pointing out that the available evidence for cultivated vines and olives suggests that they were present only in the later Bronze Age. Trade, nevertheless, was probably one of many variables that led to the emergence of palace economies in Minoan Crete. American archaeologist William Rathje developed a hypothesis that considered an explosion in long-distance exchange a fundamental cause of Mayan civilization in Mesoamerica. He suggested that the lowland Mayan environment was deficient in many vital resources, among them obsidian, salt, stone for grinding, maize, and many luxury materials. All these could be obtained from the nearby highlands, from the Valley of Mexico, and from other regions if the necessary trading networks came into being. Such connections, and the trading expeditions to maintain them, could not be organized by individual villages. The Maya lived in a relatively uniform environment, where every community suffered from the same resource deficiencies. Thus, argued Rathje, long-distance trade networks were organized through local ceremonial centers and their leaders. In time, this organization became a state, and knowledge of its functioning was exportable, as were pottery, tropical bird feathers, specialized stone materials, and other local commodities. Rathje's hypothesis probably explains part of the complex process of Mayan state formation, but it suffers from the objection that suitable alternative raw materials can be found in the lowlands. It could be, too, that warfare became a competitive response to population growth and to the increasing scarcity of prime agricultural land, and that it played an important role in the emergence of the Mayan states. Now that we know much more about ancient exchange and commerce, we know that, because no one aspect of trade was an overriding cause of cultural change or evolution in commercial practices, trade can never be looked on as a unifying factor or as a primary agent of ancient civilization. Many ever-changing variables affected ancient trade, among them the demand for goods. There were also the logistics of transportation, the extent of the trading network, and the social and political environment. Intricate market networks channeled supplies along well-defined routes. Authorities at both ends might regulate the profits fed back to the source, providing the incentive for further transactions. There may or may not have been a market organization. Extensive long-distance trade was a consequence rather than a cause of complex societies. 54-2 Overkill of the North American megafauna. Thousands of years ago, in North America's past, all of its megafauna, large mammals such as mammoths and giant bears, disappeared. One proposed explanation for this event is that when the first Americans migrated over from Asia, they hunted the megafauna to extinction. These people, known as the Clovis Society after a site where their distinctive spear points were first found, would have been able to use this food source to expand their population and fill the continent rapidly. Yet many scientists argue against this Pleistocene overkill hypothesis. Modern humans have certainly been capable of such drastic effects on animals, but could ancient people with little more than stone spears similarly have caused the extinction of numerous species of animals? 35 genera or groups of species suffered extinction in North America around 11,000 B. C, soon after the appearance and expansion of Paleoindians throughout the Americas. Although the climate changed at the end of the Pleistocene, warming trends had happened before. A period of massive extinction of large mammals like that seen about 11,000 years ago had not occurred during the previous 400,000 years. Despite these changes, the only apparently significant difference in the Americas 11,000 years ago was the presence of human hunters of these large mammals. Was this coincidence or cause and effect? We do not know. Ecologist Paul S. Martin has championed the model that associates the extinction of large mammals at the end of The Pleistocene with Human Predation, with researcher J. E. Mosselman. He has co-authored a work in which a computer model showed that in around 300 years, given the right conditions, a small influx of hunters into eastern Beringia 12,000 years ago could have spread across North America in a wave and wiped out game animals to feed their burgeoning population. The researchers ran the model several ways, always beginning with a population of 100 humans in Edmonton, in Alberta, Canada, at 11,500 years ago. Assuming different initial North American big game animal populations and different population growth rates for the human settlers, and varying kill rates, Mosselman and Martin derived figures of between 279 and 1,157 years from initial contact to big game extinction. Many scholars continue to support this scenario. For example, geologist Larry Eigenbrod has mapped the locations of dated Clovis sites alongside the distribution of dated sites where the remains of woolly mammoths have been found in both archaeological and purely paleontological contexts. These distributions show remarkable synchronicity. There are, however, many problems with this model. Significantly, though a few sites are quite impressive, there really is very little archaeological evidence to support it. Writing in 1982, Martin himself admitted the paucity of evidence. For example, at that point, the remains of only 38 individual mammoths had been found at Clovis sites. In the years since, few additional mammoths have been added to the list. There are still fewer than 20 Clovis sites where the remains of one or more mammoths have been recovered, a minuscule proportion of the millions that necessarily would have had to have been slaughtered within the overkill scenario. Though Martin claims the lack of evidence actually supports his model, the evidence is sparse because the spread of humans and the extinction of animals occurred so quickly. This argument seems weak. And how could we ever disprove it? As archaeologist Donald Grayson points out, in other cases where extinction resulted from the quick spread of human hunters, for example, the extinction of the moa, the large flightless bird of New Zealand, archaeological evidence in the form of remains is abundant. Grayson has also shown that the evidence is not so clear that all or even most of the large herbivores in late Pleistocene America became extinct after the appearance of Clovis. Of the 35 extinct genera, only 8 can be confidently assigned an extinction date of between 12,000 and 10,000 years ago. Many of the older genera, Grayson argues, may have succumbed before 12,000 BC, at least half a century before the Clovis showed up in the American West. 53-2 Rainforest soils. On viewing the lush plant growth of a tropical rainforest, most people would conclude that the soil beneath it is rich in nutrients. However, although rainforest soils are highly variable, they have in common the fact that abundant rainfall washes mineral nutrients out of them and into streams. This process is known as leaching. Because of rain leaching, most tropical rainforest soils have low to very low mineral nutrient content, in dramatic contrast to mineral-rich grassland soils. Tropical forest soils also often contain particular types of clays that, unlike the mineral-binding clays of temperate forest soils, do not bind mineral ions well. Aluminum is the dominant cation present in tropical soils, but plants do not require this element, and it is moderately toxic to a wide range of plants. Aluminum also reduces the availability of phosphorus, an element in high demand by plants. High moisture and temperatures speed the growth of soil microbes that decompose organic compounds, so tropical soils typically contain far lower amounts of organic materials than do other forest or grassland soils. Because organic compounds help loosen compact clay soils, hold water, and bind mineral nutrients, the relative lack of organic materials in tropical soils is deleterious to plants. Plant roots cannot penetrate far into hard clay soils, and during dry periods, the soil cannot hold enough water to supply plant needs. Because the concentration of dark-colored organic materials is low in tropical soils, they are often colored red or yellow by the presence of iron, aluminum, and manganese oxides. When dry, these soils become rock-hard. The famous Cambodian temples of Angkor Wat, which have survived for many centuries, were constructed from blocks of such hard rainforest soils. Given such poor soils, how can lush tropical forests exist? The answer is that the forest's minerals are held in its living biomass, the trees and other plants and the animals. In contrast to grasslands, where a large proportion of plant biomass is produced underground, that of tropical forests is nearly all above ground. Dead leaves, branches, and other plant parts, as well as the wastes and bodies of rainforest animals, barely reach the forest floor before they are rapidly decayed by abundant decomposers, bacterial and fungal minerals released by decay, are quickly absorbed by multitudinous shallow, fine tree feeder roots and stored in plant tissues. Many tropical rainforest plants have mycorrhizal partners whose delicate hyphae spread through great volumes of soil, from which they release and absorb minerals and ferry them back to the host plant in exchange for needed organic compounds. The fungal hyphae are able to absorb phosphorus that plant roots could not themselves obtain from the very dilute soil solutions, and fungal hyphae can transfer mineral nutrients from one forest plant to another. Consequently, tropical rainforests typically have what are known as closed nutrient systems, in which minerals are handed off from one organism to another with little leaking through to the soil. When mineral nutrients do not spend much time in the soil, they cannot be leached into streams. Closed nutrient systems have evolved in response to the leaching effects of heavy tropical rainfall. Evidence for this conclusion is that nutrient systems are more open in the richest tropical soils and tightest in the poorest soils. The growth of organisms is dependent on the availability of nutrients, none of which is more important than nitrogen. Although there is an abundant supply of nitrogen in Earth's atmosphere, it cannot be absorbed by plants unless it is fixed or combined chemically with other elements to form nitrogen compounds. Nitrogen-fixing bacteria help tropical rainforest plants cope with the poor soils there by supplying them with needed nitrogen. Many species of tropical rainforest trees belong to the legume family, which is known for associations of nitrogen-fixing bacteria within root nodules. Also, cycads produce special above-ground roots that harbor nitrogen-fixing cyanobacteria. By growing above the ground, the roots are exposed to sunlight, which the cyanobacteria require for growth. Nitrogen fixation by free-living bacteria in tropical soils is also beneficial. 48-2 Determining dinosaur diet. Determining what extinct dinosaurs ate is difficult, but we can infer some aspects of their dietary preferences. Traditionally, this information has been derived from direct evidence, such as stomach contents, and indirect evidence, such as establishing a correlation between particular body characteristics and diets of living animals and then inferring habits for dinosaurs. Animals such as house cats and dogs have large, stabbing canine teeth at the front of the mouth and smaller, equally sharp teeth farther back in their jaws. Many of these animals are also armed with sharp claws. The advantage of teeth and claws as predatory tools is obvious. Now consider animals like cows, horses, rabbits, and mice. These animals have flat teeth at the back of the jaw that are analogous to and have the same function as grindstones. Unlike the meat slicing and stabbing teeth of carnivores, the teeth of these animals grind and shred plant material before digestion. More clues exist in other parts of the skull. The jaw joint of carnivores such as dogs and cats has the mechanical advantage of being at the same level as the tooth row, allowing the jaws to close with tremendous speed and forcing the upper teeth to occlude against the lower teeth with great precision. In herbivorous animals, rapid jaw closure is less important. Because the flat teeth of herbivores work like grindstones, however, the jaws must move both side to side and front to back. The jaw joints of many advanced herbivores, such as cows, lie at a different level than the tooth row, allowing transverse tearing, shredding, and compression off plant material. If we extend such observations to extinct dinosaurs, we can infer dietary preferences, even though we cannot determine the exact diet. The duck-billed dinosaurs known as hadrosaurs are a good example of a group whose jaw joint is below the level of the tooth row, which probably helped them grind up tough, fibrous vegetation. Paleontologists would like to be much more specific about a dinosaur's diet than simply differentiating carnivore from herbivore. This finer level of resolution requires direct fossil evidence of dinosaur meals. Stomach contents are only rarely preserved, but when present, allow us to determine exactly what these animals were eating. In the stomach contents of specimens of Coelophysis are bones from juvenile animals of the same species. At one time, these were thought to represent embryonic animals, suggesting that this small dinosaur gave birth to live young rather than laying eggs. Further research indicated that the small dinosaurs were too large and too well developed to be pre-hatching young. In addition, the juveniles inside the body cavity were of different sizes. All the evidence points to the conclusion that these are the remains of prey items and that, as an adult, Coelophysis was at least in part a cannibal. Fossilized stomach contents are not restricted to carnivorous dinosaurs. In a few rare cases, most of them mummies, fossilized plant remains have been found inside the body cavity of hadrosaurs. Some paleontologists have argued that these represent stream accumulations rather than final meals. The best known of these cases is the second Edmontosaurus mummy collected by the Sternbergs. In the chest cavity of this specimen, which is housed in the Senckenberg Museum in Germany, are the fossil remains of conifer needles, twigs, seeds, and fruits. Similar finds in Corythosaurus specimens from Alberta, Canada, have also been reported, indicating that at least two kinds of late Cretaceous hadrosaurs fed on the sorts of tress that are common in today's boreal woodlands. A second form of direct evidence comes from coprolites. Several dinosaur fossil localities preserve coprolites. Coprolites yield unequivocal evidence about the dietary habits of dinosaurs. Many parts of plants and animals are extremely resistant to the digestive systems of animals and pass completely through the body with little or no alteration. Study of coprolites has indicated that the diets of some herbivorous dinosaurs were relatively diverse, while other dinosaurs appear to have been specialists, feeding on particular types of plants. The problem with inferring diets from coprolites is the difficulty in accurately associating a particular coprolite with a specific dinosaur. 47-2 Termite ingenuity. Termites, social insects which live in colonies that, in some species, contain 2 million individuals or more, are often incorrectly referred to as white ants. But they are certainly not ants. Termites, unlike ants, have gradual metamorphosis with only 3 life stage: egg, nymph, and adult. Ants and the other social members of their order, certain bees and wasps, have complete metamorphosis in 4 life stages: egg, larva, pupa, and adult. The worker and soldier castes of social ants, bees, and wasps consist of only females, all daughters of a single queen that mated soon after she matured and thereafter never mated again. The worker and soldier castes of termites consist of both males and females, and the queen lives permanently with a male consort. Since termites are small and soft-bodied, they easily become desiccated and must live in moist places with a high relative humidity. They do best when the relative humidity in their nest is above 96% and the temperature is fairly high, an optimum of about 79 degrees Fahrenheit for temperate zone species and about 86 degrees Fahrenheit for tropical species. Subterranean termites, the destructive species that occurs commonly throughout the eastern United States, attain these conditions by nesting in moist soil that is in contact with wood, their only food. The surrounding soil keeps the nest moist and tends to keep the temperature at a more or less favorable level. When it is cold in winter, subterranean termites move to burrows below the frost line. Some tropical termites are more ingenious engineers, constructing huge above-ground nests with built-in air conditioning that keeps the nest moist, at a constant temperature, and well supplied with oxygen. Among the most architecturally advanced of these termites is an African species, Macrotermes natalensis. Renowned Swiss entomologist Martin Luscher described the mounds of this fungus-growing species as being as much as 16 feet tall, 16 feet in diameter at their base, and with a cement-like wall of soil mixed with termite saliva that is from 16 to 23 inches thick. The thick and dense wall of the mound insulates the interior microclimate from the variations in humidity and temperature of the outside atmosphere. Several narrow and relatively thin-walled ridges on the outside of the mound extend from near its base almost to its top. According to Luscher, a medium-sized nest of macrotermes has a population of about 2 million individuals. The metabolism of so many termites and of the fungus that they grow in their gardens as food helps keep the interior of the nest warm and supplies some moisture to the air in the nest. The termites saturate the atmosphere of the nest, bringing it to about 100% relative humidity, by carrying water up from the soil. But how is this well-insulated nest ventilated? Its many occupants require over 250 quarts of oxygen per day. How can so much oxygen diffuse through the thick walls of the mound? Even the pores in the wall are filled with water, which almost stops the diffusion of gases. The answer lies in the construction of the nest. The interior consists of a large central core in which the fungus is grown, below it is cellar of empty space, above it is an attic of empty space, and within the ridges on the outer wall of the nest, there are many small tunnels that connect the cellar and the attic. The warm air in the fungus gardens rises through the nest up to the attic. From the attic, the air passes into the tunnels in the ridges and flows back down to the cellar. Gases, mainly oxygen coming in and carbon dioxide going out, easily diffuse into or out of the ridges, since their walls are thin and their surface area is large because they protrude far out from the wall of the mound. Thus air that flows down into the cellar through the ridges is relatively rich in oxygen, and has lost much of its carbon dioxide. It supplies the nest's inhabitants with fresh oxygen as it rises through the fungus growing area back up to the attic. 47-3 Coral reefs. An important environment that is more or less totally restricted to the intertropical zone is the coral reef. Coral reefs are found where the ocean water temperature is not less than 21 degrees Celsius, where there is a firm substratum, and where the seawater is not rendered too dark by excessive amounts of river-borne sediment. They will not grow in very deep water, so a platform within 30 to 40 meters of the surface is a necessary prerequisite for their development. Their physical structure is dominated by the skeletons of corals, which are carnivorous animals living off zooplankton. However, in addition to corals there are enormous quantities of algae, some calcareous, which help to build the reefs. The size of reefs is variable. Some atolls are very large. Kwajalein in the Marshall Islands of the South Pacific is 120 kilometers long and as much as 24 kilometers across, but most are very much smaller, and rise only a few meters above the water. The 2,000-kilometer complex of reefs known as the Great Barrier Reef, which forms a gigantic natural breakwater off the northeast coast of Australia, is by far the greatest coral structure on Earth. Coral reefs have fascinated scientists for almost 200 years, and some of the most pertinent observations of them were made in the 1830s by Charles Darwin on the voyage of the Beagle. He recognized that there were three major kinds: fringing reefs, barrier reefs, and atolls, and he saw that they were related to each other in a logical and gradational sequence. A fringing reef is one that lies close to the shore of some continent or island. Its surface forms an uneven and rather rough platform around the coast, about the level of low water, and its outer edge slopes downwards into the sea. Between the fringing reef and the land there is sometimes a small channel or lagoon. When the lagoon is wide and deep and the reef lies at some distance from the shore and rises from deep water, it is called a barrier reef. An atoll is a reef in the form of a ring or horseshoe with a lagoon in the center. Darwin's theory was that the succession from one coral reef type to another could be achieved by the upward growth of coral from a sinking platform, and that there would be a progression from a fringing reef through the barrier reef stage until, with the disappearance through subsidence of the central island, only a reef-enclosed lagoon or atoll would survive. A long time after Darwin put forward this theory, some deep boreholes were drilled in the Pacific atolls in the 1950s. The drill holes passed through more than 1,000 meters of coral before reaching the rock substratum of the ocean floor, and indicated that the coral had been growing upward for tens of millions of years as Earth's crust subsided at a rate of between 15 and 51 meters per million years. Darwin's theory was therefore proved basically correct. There are some submarine islands called geos and seamounts, in which subsidence associated with seafloor spreading has been too speedy for coral growth to keep up. Like mangrove swamps, coral reefs are extremely important habitats. Their diversity of coral genera is greatest in the warm waters of the Indian Ocean and the western Pacific. Indeed, they have been called the marine version of the tropical rainforest, rivaling their terrestrial counterparts in both richness of species and biological productivity. They also have significance because they provide coastal protection, opportunities for recreation, and are potential sources of substances like medicinal drugs. At present they are coming under a variety of threats, of which two of the most important are dredging and the effects of increased siltation brought about by accelerated erosion from neighboring land areas. 46-3 Ecosystem diversity and stability. Conservation biologists have long been concerned that species extinction could have significant consequences for the stability of entire ecosystems, groups of interacting organisms, and the physical environment that they inhabit. An ecosystem could survive the loss of some species, but if enough species were lost, the ecosystem would be severely degraded. In fact, it is possible that the loss of a single important species could start a cascade of extinctions that might dramatically change an entire ecosystem. A good illustration of this occurred after sea otters were eliminated from some Pacific kelp bed ecosystems. The kelp beds were practically obliterated too because in the absence of sea otter predation, sea urchin populations exploded and consumed most of the kelp and other macroalgae. It is usually claimed that species-rich ecosystems tend to be more stable than species-poor ecosystems. 3 mechanisms by which higher diversity increases ecosystem stability have been proposed. First, if there are more species in an ecosystem, then its food web will be more complex, with greater redundancy among species in terms of their nutritional roles. In other words, in a rich system, if a species is lost, there is a good chance that other species will take over its function as prey, predator, producer, decomposer, or whatever role it played. Second, diverse ecosystems may be less likely to be invaded by new species, notably exotics, that would disrupt the ecosystem structure and function. Third, in a species-rich ecosystem, diseases may spread more slowly because most species will be relatively less abundant, thus increasing the average distance between individuals of the same species and hampering disease transmission among individuals. Scientific evidence to illuminate these ideas has been slow in coming, and many shadows remain. One of the first studies to provide data supporting a relationship between diversity and stability examined how grassland plants responded to a drought. Researchers D. Tillman and J. A. Downing used the ratio of above-ground biomass in 1988 to that in 1986 in 207 plots in a grassland field in the Cedar Creek Natural History Area in Minnesota as an index of ecosystem response to disruption by drought. In an experiment that began in 1982, they compared these values with the number of plant species in each plot and discovered that the plots with a greater number of plant species experienced a less dramatic reduction in biomass. Plots with more than 10 species had about half as much biomass in 1988 as in 1986, whereas those with fewer than 5 species only produced roughly 1/8 as much biomass after the 2-year drought. Apparently, species-rich plots were likely to contain some drought-resistant plant species that grew better in drought years, compensating for the poor growth of less tolerant species. To put this result in more general terms, a species-rich ecosystem may be more stable because it is more likely to have species with a wide array of responses to variable conditions such as droughts. Furthermore, a species-rich ecosystem is more likely to have species with similar ecological functions, so that if a species is lost from an ecosystem, another species, probably a competitor, is likely to flourish and occupy its functional role. Both of these, variability in responses and functional redundancy, could be thought of as insurance against disturbances. The Minnesota grassland research has been widely accepted as strong evidence for the diversity stability theory. However, its findings have been questioned, and similar studies on other ecosystems have not always found a positive relationship between diversity and stability. Clearly, this is a complex issue that requires further field research with a broad spectrum of ecosystems and species. Grassland plants and computer models will only take us so far. In the end, despite insightful attempts to detect some general patterns, we may find it very difficult to reduce this topic to a simple, universal truth. 45:1 microscopes. The Beringia landscape during the peak of the last ice age, Northeast Asia and Alaska were connected by a broad land mass called the Bering Land Bridge. This land bridge existed because so much of Earth's water was frozen in the great ice sheets that sea levels were over 100 meters lower than they are today. Between 25,000 and 10,000 years ago, Siberia, the Bering Land Bridge, and Alaska shared many environmental characteristics. These included a common mammalian fauna of large mammals, a common flora composed of broad grasslands as well as windswept dunes and tundra, and a common climate with cold, dry winters and somewhat warmer summers. The recognition that many aspects of the modern flora and fauna were present on both sides of the Bering Sea as remnants of the Ice Age landscape led to this region being named Beringia. It is through Beringia that small groups of large mammal hunters, slowly expanding their hunting territories, eventually colonized North and South America. On this archaeologists generally agree, but that is where the agreement stops. One broad area of disagreement in explaining the peopling of the Americas is the domain of paleoecologists, but it is critical to understanding human history. What was Beringia like? The Beringian landscape was very different from what it is today. Broad, windswept valleys, glaciated mountains, sparse vegetation, and less moisture created a rather forbidding land mass. This land mass supported herds of now extinct species of mammoth, bison, and horse and somewhat modern versions of caribou, muskox, elk, and saiga antelope. These grazers supported in turn a number of impressive carnivores, including the giant short-faced bear, the saber-toothed cat, and a large species of lion. The presence of mammal species that require grassland vegetation has led Arctic biologist Dale Guthrie to argue that while cold and dry, there must have been broad areas of dense vegetation to support herds of mammoth, horse, and bison. Further, nearly all of the Ice Age fauna had teeth that indicate an adaptation to grasses and sedges. They could not have been supported by a modern flora of mosses and lichens. Guthrie has also demonstrated that the landscape must have been subject to intense and continuous winds, especially in winter. He makes this argument based on the anatomy of horse and bison, which do not have the ability to search for food through deep snow cover. They need landscapes with strong winds that remove the winter snows, exposing the dry grasses beneath. Guthrie applied the term mammoth steppe to characterize this landscape. In contrast, Paul Kallenbachs has offered a counterargument based on the analysis of pollen in lake sediments dating to the last ice age. He found that the amount of pollen recovered in these sediments is so low that the Beringian landscape during the peak of the last glaciation was more likely to have been what he termed a polar desert, with little or only sparse vegetation. In no way was it possible that this region could have supported large herds of mammals and thus, human hunters. Guthrie has argued against this view by pointing out that radiocarbon analysis of mammoth, horse, and bison bones from Beringian deposits revealed that the bones date to the period of most intense glaciation. The argument seemed to be at a standstill until a number of recent studies resulted in a spectacular suite of new finds. The first was the discovery of a 1,000 square kilometer preserved patch of Beringian vegetation dating to just over 17,000 years ago, the peak of the last ice age. The plants were preserved under a thick ash fall from a volcanic eruption. Investigations of the plants found grasses, sedges, mosses, and many other varieties in a nearly continuous cover, as was predicted by Guthrie. But this vegetation had a thin root mat with no soil formation, demonstrating that there was little long-term stability in plant cover, a finding supporting some of the arguments of Kallenbachs. A mixture of continuous but thin vegetation supporting herds of large mammals is one that seems plausible and realistic with the available data. 45-2 Wind pollination. Pollen, a powdery substance which is produced by flowering plants and contains male reproductive cells, is usually carried from plant to plant by insects or birds, but some plants rely on the wind to carry their pollen. Wind pollination is often seen as being primitive and wasteful in costly pollen, and yet it is surprisingly common, especially in higher latitudes. Wind is very good at moving pollen a long way. Pollen can be blown for hundreds of kilometers, and only birds can get pollen anywhere near as far. The drawback is that wind is obviously unspecific as to where it takes the pollen. It is like trying to get a letter to a friend at the other end of the village by climbing onto the roof and throwing an armful of letters into the air and hoping that one will end up in the friend's garden. For the relatively few dominant tree species that make up temperate forests, where there are many individuals of the same species within pollen range, this is quite a safe gamble. If a number of people in the village were throwing letters off roofs, your friend would be bound to get one. By contrast, in the tropics, where each tree species has few, widely scattered individuals, the chance of wind blowing pollen to another individual is sufficiently slim that animals are a safer bet as transporters of pollen. Even tall trees in the tropics are usually not wind pollinated despite being in windy conditions. In a similar way, trees in temperate forests that are insect pollinated tend to grow as solitary, widely spread individuals. Since wind-pollinated flowers have no need to attract insects or other animals, they have dispensed with bright petals, nectar, and scent. These are at best a waste and at worst an impediment to the transfer of pollen in the air. The result is insignificant-looking flowers and catkins. Wind pollination does, of course, require a lot of pollen. Birch and hazel trees can produce 5.5 and 4 million grains per catkin, respectively. There are various adaptations to help as much of the pollen go as far as possible. Most deciduous wind-pollinated Trees produce their pollen in the spring while the branches are bare of leaves to reduce the surrounding surfaces that compete with the stigmas for pollen. Evergreen conifers, which do not shed their leaves, have less to gain from spring flowering, and, indeed, some flower in the autumn or winter. Pollen produced higher in the top branches is likely to go farther. It is windier and the pollen can be blown farther before hitting the ground. Moreover, dangling catkins like hazel hold the pollen in until the wind is strong enough to bend them, ensuring that pollen is only shed into the air when the wind is blowing hard. Weather is also important. Pollen is shed primarily when the air is dry to prevent too much sticking to wet surfaces or being knocked out of the air by rain. Despite these adaptations, much of the pollen fails to leave the top branches, and only between 0.5% and 40% gets more than 100 meters away from the parent. But once this far, significant quantities can go a kilometer or more. Indeed, pollen can travel many thousands of kilometers at high altitudes. Since all this pollen is floating around in the air, it is no wonder that wind-pollinated trees are a major source of allergies. Once the pollen has been snatched by the wind, the fate of the pollen is obviously up to the vagaries of the wind, but not everything is left to chance. Wind-borne pollen is dry, rounded, smooth, and generally smaller than that of insect-pollinated plants. But size is a two-edged sword. Small grains may be blown farther, but they are also more prone to be whisked past the waiting stigma because smaller particles tend to stay trapped in the fast-moving air that flows around the stigma. But stigmas create turbulence, which slows the airspeed around them and may help pollen stick to them. 45-3 Feeding strategies in the ocean. In the open sea, animals can often find food reliably available in particular regions or seasons. In these circumstances, animals are neither constrained to get the last calorie out of their diet nor is energy conservation a high priority. In contrast, the food levels in the deeper layers of the ocean are greatly reduced, and the energy constraints on the animals are much more severe. To survive at those levels, animals must maximize their energy input, finding and eating whatever potential food source may be present. In the near-surface layers, there are many large, fast carnivores as well as an immense variety of planktonic animals, which feed on plankton by filtering them from currents of water that pass through a specialized anatomical structure. These filter feeders thrive in the well-illuminated surface waters because oceans have so many very small organisms, from bacteria to large algae to larval crustaceans. Even fishes can become successful filter feeders in some circumstances. Although the vast majority of marine fishes are carnivores, in near-surface regions of high productivity the concentrations of larger phytoplankton are sufficient to support huge populations of filter-feeding sardines and anchovies. These small fishes use their gill filaments to strain out the algae that dominate such areas. Sardines and anchovies provide the basis for huge commercial fisheries as well as a food resource for large numbers of local carnivores, particularly seabirds. At a much larger scale, baleen whales and whale sharks are also efficient filter feeders in productive coastal or polar waters, although their filtered particles comprise small animals such as copepods and krill rather than phytoplankton. Filtering seawater for its particulate nutritional content can be an energetically demanding method of feeding, particularly when the current of water to be filtered has to be generated by the organism itself, as is the case for all planktonic animals, particulate organic matter of at least 2.5 micrograms per cubic liter is required to provide a filter-feeding planktonic organism with a net energy gain. This value is easily exceeded in most coastal waters, but in the deep sea, the levels of organic matter range from next to nothing to around 7 micrograms per cubic liter. Even though mean levels may mask much higher local concentrations, it is still the case that many deep-sea animals are exposed to conditions in which a normal filter feeder would starve. There are, therefore, fewer successful filter feeders in deep water, and some of those that are there have larger filtering systems to cope with the scarcity of particles. Another solution for such animals is to forage in particular layers of water where the particles may be more concentrated. Many of the groups of animals that typify the filter-feeding lifestyle in shallow water have deep-sea representatives that have become predatory. Their filtering systems, which reach such a high degree of development in shallow water species, are greatly reduced. Alternative methods of active or passive prey capture have been evolved, including trapping and seizing prey, entangling prey, and sticky tentacles. In the deeper waters of the oceans, there is a much greater tendency for animals to await the arrival of food particles or prey, rather than to search them out actively. This has resulted in a more stealthy style of feeding, with the consequent emphasis on lures and/or the evolution of elongated appendages that increase the active volume of water controlled or monitored by the animal. Another consequence of the limited availability of prey is that many animals have developed ways of coping with much larger food particles, relative to their own body size, than the equivalent shallower species can process. Among the fishes, there is a tendency for the teeth and jaws to become appreciably enlarged. In such creatures, not only are the teeth hugely enlarged and/or the jaws elongated, but the size of the mouth opening may be greatly increased by making the jaw articulations so flexible that they can be effectively dislocated. Very large or long teeth provide almost no room for cutting the prey into a convenient size for swallowing. The fish must gulp the prey down whole. 44:1 from fish to terrestrial vertebrates. One of the most significant evolutionary events that occurred on Earth was the transition of water-dwelling fish to terrestrial tetrapods. Fish probably originated in the oceans, and our first records of them are in marine rocks. However, by the Devonian period, they had radiated into almost all available aquatic habitats, including freshwater settings. One of the groups whose fossils are especially common in rocks deposited in fresh water is the lobefinned fish. The freshwater Devonian lobefinned fish Rhipidistion crossopterygian is of particular interest to biologists studying tetrapod evolution. These fish lived in river channels and lakes on large deltas. The delta rocks in which these fossils are found are commonly red due to oxidized iron minerals, indicating that the deltas formed in a climate that had alternate wet and dry periods. If there were periods of drought, any adaptations allowing the fish to survive the dry conditions would have been advantageous. In these Rhipidistions, several such adaptations existed. It is known that they had lungs as well as gills for breathing. Cross-sections cut through some of the fossils reveal that the mud filling the interior of the carcass differed in consistency and texture depending on its location inside the fish. These differences suggest a sac-like cavity below the front end of the gut that can only be interpreted as a lung. Gills were undoubtedly the main source of oxygen for these fish, but the lungs served as an auxiliary breathing device for gulping air when the water became oxygen depleted, such as during extended periods of drought. These fish had already evolved one of the prime requisites for living on land, the ability to use air as a source of oxygen. A second adaptation of these fish was in the structure of the lobe fins. The fins were thick, fleshy, and quite sturdy, with a median axis of bone down the center. They could have been used as feeble locomotor devices on land, perhaps good enough to allow a fish to flop its way from one pool of water that was almost dry to an adjacent pond that had enough water and oxygen for survival. These fins eventually changed into short, stubby legs. The bones of the fins of a Devonian Rhipidistion exactly match in number and position the limb bones of the earliest known tetrapods, the amphibians. It should be emphasized that the evolution of lungs and limbs was in no sense an anticipation of future life on land. These adaptations developed because they helped fish to survive in their existing aquatic environment. What ecological pressures might have caused fishes to gradually abandon their watery habitat and become increasingly land-dwelling creatures? Changes in climate during the Devonian may have had something to do with this if freshwater areas became progressively more restricted. Another impetus may have been new sources of food. The edges of ponds and streams surely had scattered dead fish and other water-dwelling. In addition, plants had emerged into terrestrial habitats in areas near streams and ponds, and crabs and other arthropods were also members of this earliest terrestrial community. Thus, by the Devonian, the land habitat marginal to freshwater was probably a rich source of protein that could be exploited by an animal that could easily climb out of water. Evidence from teeth suggests that these earliest tetrapods did not utilize land plants as food. They were presumably carnivorous and had not developed the ability to feed on plants. How did the first tetrapods make the transition to a terrestrial habitat? Like early land plants such as rhiniophytes, they made only a partial transition. They were still quite tied to water. However, many problems that faced early land plants were not applicable to the first tetrapods. The ancestors of these animals already had a circulation system, and they were mobile, so that they could move to water to drink. Furthermore, they already had lungs, which ripidistions presumably used for auxiliary breathing. The principal changes for the earliest tetrapods were in the skeletal system, changes in the bones of the fins, the vertebral column, pelvic girdle, and pectoral girdle. 44-3 Seagrasses. Many areas of the shallow sea bottom are covered with a lush growth of aquatic flowering plants adapted to live submerged in seawater. These plants are collectively called seagrasses. Seagrass beds are strongly influenced by several physical factors. The most significant is water motion, currents, and waves. Since seagrass systems exist in both sheltered and relatively open areas, they are subject to differing amounts of water motion. For any given seagrass system, however, the water motion is relatively constant. Seagrass meadows in relatively turbulent waters tend to form a mosaic of individual mounds, whereas meadows in relatively calm waters tend to form flat, extensive carpets. The seagrass beds, in turn, dampen wave action, particularly if the blades reach the water surface. This damping effect can be significant to the point where just 1 meter into a seagrass bed the wave motion can be reduced to zero. Currents are also slowed as they move into the bed. The slowing of wave action and currents means that seagrass beds tend to accumulate sediment. However, this is not universal and depends on the currents under which the bed exists. Seagrass beds under the influence of strong currents tend to have many of the lighter particles, including seagrass debris, moved out, whereas beds in weak current areas accumulate lighter detrital material. It is interesting that temperate seagrass beds accumulate sediments from sources outside the beds, whereas tropical seagrass beds derive most of their sediments from within. Since most seagrass systems are depositional environments, they eventually accumulate organic material that leads to the creation of fine-grained sediments with a much higher organic content than that of the surrounding unvegetated areas. This accumulation, in turn, reduces the water movement and the oxygen supply. The high rate of metabolism of the microorganisms in the sediments causes sediments to be anaerobic below the first few millimeters, according to ecologist J. W. Kenworthy. Anaerobic processes of the microorganisms in the sediment are an important mechanism for regenerating and recycling nutrients and carbon, ensuring the high rates of productivity, that is, the amount of organic material produced that are measured in those beds. In contrast to other productivity in the ocean, which is confined to various species of algae and bacteria dependent on nutrient concentrations in the water column, seagrasses are rooted plants that absorb nutrients from the sediment or substrate. They are, therefore, capable of recycling nutrients into the ecosystem that would otherwise be trapped in the bottom and rendered unavailable. Other physical factors that have an effect on seagrass beds include light, temperature, and desiccation. For example, water depth and turbidity together or separately control the amount of light available to the plants and the depth to which the seagrasses may extend. Although marine botanist W. Odsechel suggested early on that temperature was critical to the growth and reproduction of eelgrass, it has since been shown that this particularly widespread seagrass grows and reproduces at temperatures between 2 and 4 degrees Celsius in the Arctic and at temperatures up to 28 degrees Celsius on the northeastern coast of the United States. Still, extreme temperatures, in combination with other factors, may have dramatic detrimental effects. For example, in areas of the cold North Atlantic, ice may form in winter. Researchers Robertson and Mann note that when the ice begins to break up, the wind and tides may move the ice around, scouring the bottom and uprooting the eelgrass. In contrast, at the southern end of the eelgrass range, on the southeastern coast of the United States, temperatures over 30 degrees Celsius in summer cause excessive mortality. Seagrass beds also decline if they are subjected to too much exposure to the air. The effect of desiccation is often difficult to separate from the effect of temperature. Most seagrass beds seem tolerant of considerable changes in salinity and can be found in brackish waters as well as in full-strength seawater. 42-1 Geographic isolation of species. Biologist Ernst Mayr defined a species as an actually or potentially interbreeding population that does not interbreed with other such populations when there is opportunity to do so. A key event in the origin of many species is the separation of a population with its gene pool from other populations of the same species, thereby preventing population interbreeding. With its gene pool isolated, a separate population can follow its own evolutionary course. In the formation of many species, the initial isolation of a population seems to have been a geographic barrier. This mode of evolving new species is called allopatric speciation. Many factors can isolate a population geographically. A mountain range may emerge and gradually split a population of organisms that can inhabit only lowland lakes. Certain fish populations might become isolated in this way. Similarly, a creeping glacier may gradually divide a population, or a land bridge such as the Isthmus of Panama may form and separate the marine life in the ocean waters on either side. How formidable must a geographic barrier be to keep populations apart? It depends on the ability of the organisms to move across barriers. Birds and coyotes can easily cross mountains and rivers. The passage of wind-blown tree pollen is also not hindered by such barriers, and the seeds of many plants may be carried back and forth on animals. In contrast, small rodents may find a deep canyon or a wide river an effective barrier. For example, the Grand Canyon, in the southwestern United States, separate the range of the white-tailed antelope squirrel from that of the closely related Harris's antelope squirrel. Smaller, with a shorter tail that is white underneath, the white-tailed antelope squirrel inhabits deserts north of the canyon and west of the Colorado River in Southern California. Harris's antelope squirrel has a more limited range in deserts south of the Grand Canyon. Geographic isolation creates opportunities for new species to develop, but it does not necessarily lead to new species because speciation occurs only when the gene pool undergoes enough changes to establish reproductive barriers between the isolated population and its parent population. The likelihood of allopatric speciation increases when a population is small as well as isolated, making it more likely than a large population to have its gene pool changed substantially. For example, in less than 2 million years, small populations of stray animals and plants from the South American mainland that managed to colonize the Galapagos Islands gave rise to all the species that now inhabit the islands. When oceanic islands are far enough apart to permit populations to evolve in isolation, but close enough to allow occasional dispersions to occur, they are effectively outdoor laboratories of evolution. The Galapagos Island chain is one of the world's greatest showcases of evolution. Each island was born from underwater volcanoes and was gradually covered by organisms derived from strays that rode the ocean currents and winds from other islands and continents. Organisms can also be carried to islands by other organisms, such as sea birds that travel long distances with seeds clinging to their feathers. The species on the Galapagos Islands today, most of which occur nowhere else, descended from organisms that floated, flew, or were blown over the sea from the South American mainland. For instance, the Galapagos Island chain has a total of 13 species of closely related birds called Galapagos finches. These birds have many similarities but differ in their feeding habits and their beak type, which is correlated with what they eat. Accumulated evidence indicates that all 13 finch species evolved from a single small population of ancestral birds that colonized one of the islands. Completely isolated on the island after migrating from the mainland, the founder population may have undergone significant changes in its gene pool and become a new species. Later, a few individuals of this new species may have been blown by storms to a neighboring island. Isolated on this second island, the second founder population could have evolved into a second new species, which could later recolonize the island from which its founding population emigrated. Today, each Galapagos island has multiple species of finches, with as many as 10 on some islands. 42-2 Explaining dinosaur extinction. Dinosaurs rapidly became extinct about 65 million years ago as part of a mass extinction known as the KT event, because it is associated with a geological signature known as the KT boundary, usually a thin band of sedimentation found in various parts of the world. Many explanations have been proposed for why dinosaurs became extinct. For example, some have blamed dinosaur extinction on the development of flowering plants, which were supposedly more difficult to digest and could have caused constipation or indigestion, except that flowering plants first evolved in the early Cretaceous, about 60 million years before the dinosaurs died out. In fact, several scientists have suggested that the duckbill dinosaurs and horned dinosaurs, with their complex battery of grinding teeth, evolved to exploit this new resource of rapidly growing flowering plants. Others have blamed extinction on competition from the mammals, which allegedly ate all the dinosaur eggs, except that mammals and dinosaurs appeared at the same time in the late Triassic, about 190 million years ago, and there is no reason to believe that mammals suddenly acquired a taste for dinosaur eggs after 120 million years of coexistence. Some explanations fail because there is no way to scientifically test them, and they cannot move beyond the realm of speculation and guesswork. This focus on explaining dinosaur extinction misses an important point. The extinction at the end of the Cretaceous was a global event that killed off organisms up and down the food chain. It wiped out many kinds of plankton in the ocean and many marine organisms that lived on the plankton at the base of the food chain. These included a variety of clams and snails, and especially the ammonites, a group of shelled squid-like creatures that dominated the Mesozoic seas and had survived many previous mass extinctions. The KT event marked the end of the marine reptiles, such as the mosasaurs and the plesiosaurs, which were the largest creatures that had ever lived in the seas and which ruled the seas long before whales evolved. On land, there was also a crisis among the land plants, in addition to the disappearance of dinosaurs. So any event that can explain the destruction of the base of the food chain can better explain what happened to organisms at the top of the food chain, such as the dinosaurs. By contrast, any explanation that focuses strictly on the dinosaurs completely misses the point. The Cretaceous extinctions were a global phenomenon, and dinosaurs were just a part of a bigger picture. According to one theory, the age of dinosaurs ended suddenly 65 million years ago when a giant rock from space plummeted to Earth. Estimated to be 10 to 15 kilometers in diameter, this bolide was traveling at cosmic speeds of 20 to 70 kilometers per second, or 45,000 to 156,000 miles per hour. Such a huge mass traveling at such tremendous speeds carries an enormous amount of energy. When the bolide struck, this energy was released and generated a huge shock wave that leveled everything for thousands of kilometers around the impact and caused most of the landscape to burst into flames. The bolide struck an area of the Yucatan Peninsula of Mexico known as Chicxulub, excavating a crater 15 to 20 kilometers deep and at least 170 kilometers in diameter. The impact displaced huge volumes of seawater, causing much flood damage in the Caribbean. Meanwhile, the bolide itself excavated 100 cubic kilometers of rock and debris from the site, which rose to an altitude of 100 kilometers. Most of it fell back immediately, but some of it remained as dust in the atmosphere for months. This material, along with the smoke from the fires, shrouded Earth, creating a form of nuclear winter. According to computerized climate models, global temperatures fell to near the freezing point, photosynthesis halted, and most plants on land and in the sea died. With the bottom of the food chain destroyed, dinosaurs could not survive. 42 latitude in biodiversity. When we look at the way in which biodiversity is distributed over the land surface of the planet, we find that it is far from even. The tropics contain many more species overall than an equivalent area at the higher latitudes. This seems to be true for many different groups of animals and plants. Why is it that higher latitudes have lower diversities than the tropics? Perhaps it is simply a matter of land area. The tropics contain a larger surface area of land than higher latitudes, a fact that is not always evident when we examine commonly used projections of Earth's curved surface, since this tends to exaggerate the areas of land in the higher latitudes, and some biogeographers regard the differences in diversity as a reflection of this effect. But an analysis of the data by biologist Klaus Rohde does not support this explanation. Although area may contribute to biodiversity, it is certainly not the whole story, otherwise, large landmasses would always be richer in species. Productivity seems to be involved instead, though perhaps its influence is indirect. Where conditions are most suitable for plant growth, that is, where temperatures are relatively high and uniform and where there is an ample supply of water, one usually finds large masses of vegetation. This leads to a complex structure in the layers of plant material in a tropical rainforest. For example, a very large quantity of plant material builds up above the surface of the ground. There is also a large mass of material developed below ground as root tissues, but this is less apparent. Careful analysis of the above-ground material reveals that it is arranged in a series of layers, the precise number of layers varying with age and the nature of the forest. The arrangement of the biological mass. of the vegetation into layered forms is termed its structure. Structure is essentially the architecture of vegetation, and as in the case of tropical forests, it can be extremely complicated. In a mature floodplain tropical forest in the Amazon River basin, the canopy takes on a stratified structure. There are 3 clear peaks in leaf cover at heights of approximately 3, 6, and 30 meters above the ground, and the very highest layer, at 50 meters, corresponds to the very tall trees that stand free of the main canopy and form an open layer of their own. So, such a forest contains essentially 4 layers of canopy. Forests in temperate lands often have just 2 canopy layers, so they have much less complex architecture. Structure has a strong influence on the animal life inhabiting a site. It forms the spatial environment within which an animal feeds, moves around, shelters, lives, and breeds. It even affects the climate on a very local level by influencing light intensity, humidity, and both the range and extremes of temperature. An area of grassland vegetation with very simple structure, for example, has a very different microclimate at the ground level from that experienced in the upper canopy. Wind speeds are lower, temperatures are lower during the day, and the relative humidity is much greater near the ground. The complexity of the microclimate is closely related to the complexity of structure in vegetation, and generally speaking, the more complex the structure of vegetation, the more species of animal are able to make a living there. The high plant biomass of the tropics leads to a greater spatial complexity in the environment, and this leads to a higher potential for diversity in the living things that can occupy a region. The climates of the higher latitudes are generally less favorable for the accumulation of large quantities of biomass, hence, the structure of vegetation is simpler and the animal diversity is consequently lower. 40-3 Amphibian thermoregulation. In contrast to mammals and birds, amphibians are unable to produce thermal energy through their metabolic activity, which would allow them to regulate their body temperature independent of the surrounding or ambient temperature. However, the idea that amphibians have no control whatsoever over their body temperature has been proven false because their body temperature does not always correspond to the surrounding temperature. While amphibians are poor thermoregulators, they do exercise control over their body temperature to a limited degree. Physiological adaptations can assist amphibians in colonizing habitats where extreme conditions prevail. The tolerance range in body temperature represents the range of temperatures within which a species can survive. One species of North American newt is still active when temperatures drop to -2 degrees Celsius, while one South American frog feels comfortable even when temperatures measured to 41 degrees Celsius, the highest body temperature measured in a free-ranging amphibian. Recently it has been shown that some North American frog and toad species can survive up to 5 days with a body temperature of -6 degrees Celsius with approximately 1/3 of their body fluids frozen. The other tissues are protected because they contain the frost protective agents glycerin or glucose. Additionally, in many species the tolerance boundaries are flexible and can change as a result of acclimatization. Frog species that remain exposed to the sun despite high diurnal temperatures exhibit some fascinating modifications in the skin structure that function as morphological adaptations. Most amphibian skin is fully water permeable and is therefore not a barrier against evaporation or solar radiation. The African savanna frog Hyperoleus viridiflavus stores guanine crystals in its skin, which enable it to better reflect solar radiation, thus providing protection against overheating. The tree frog Philomedusa sauvageira responds to evaporative losses with gland secretions that provide a greasy film over its entire body that helps prevent desiccation. However, behavior is by far the most important factor in thermoregulation. The principal elements in behavioral thermoregulation are basking, heat exchange with substrates such as rock or earth, and diurnal and annual avoidance behaviors, which include moving to shelter during the day for cooling and hibernating or estivating. Heliothermy is especially common among frogs and toads. It allows them to increase their body temperature by more than 10 degrees Celsius. The Andean toad Bufo spinulosus exposes itself immediately after sunrise on moist ground and attains its preferred body temperature by this means, long before either ground or air is correspondingly warmed. A positive side effect of this approach is that it accelerates the digestion of the prey consumed overnight, thus also accelerating growth. Thigmothermy is a behavior present in most amphibians, although pressing against the ground serves a dual purpose: heat absorption by conductivity and water absorption through the skin. The effect of thigmothermy is especially evident in the Andean toad during rainfall. Its body temperature corresponds to the temperature of the warm earth and not to the much cooler air temperature. Avoidance behavior occurs whenever physiological and morphological adaptations are insufficient to maintain body temperature within the vital range. Nocturnal activity in amphibians with low tolerance for high ambient temperatures is a typical thermoregulatory behavior of avoidance. Seasonal avoidance behavior is extremely important in many amphibians. Species whose habitat lies in the temperate latitudes are confronted by lethal low temperatures in winter, while species dwelling in semi-arid regions are exposed to long dry, hot periods in summer. In amphibians, hibernation occurs in mud or deep holes away from frost. North of the Pyrenees Mountains, the natterjack toad offers a good example of hibernation, passing the winter dug deep into sandy ground. Conversely, natterjacks in southern Spain remain active during the mild winters common to the region and are instead forced into inactivity during the dry, hot summer season. Summer estivation also occurs by burrowing into the ground or hiding in cool, deep rock crevasses to avoid desiccation and lethal ambient temperature. Amphibians are therefore hardly at mercy of ambient temperature, since by means of the mechanisms described above they are more than exercise some control over their body temperature. 39-2 The extinction of moa. Between 80 and 85 million years ago, Gondwanaland, a giant continent made up of what today is Africa, Antarctica, Australia, and South America, broke up, thus causing what is now New Zealand to become separated from the larger landmass. After the separation, any creature unable to cross a considerable distance of ocean could not migrate to New Zealand. Snakes and most mammals evolved after the separation. Thus there are no New Zealand snakes, and bats, which flew there, and seals, which swam there, were the only mammals on New Zealand when Polynesian settlers arrived there about 1,000 years ago. When the Maori arrived in New Zealand, they encountered birds that had been evolving for 80 million years without the presence of mammalian predators. The most striking of these animals must have been moa. Now extinct, moa were gigantic wingless birds that stood as much as 10 feet tall and weighed as much as 550 pounds. They are known from a diverse array of remains including eggshells, eggs, a few mummified carcasses, vast numbers of bones, and some older fossilized bone. The species of moa that are currently recognized occupied ecological niches customarily filled elsewhere by large mammalian browsing herbivores. They may have had relatively low reproductive rates. Apparently, they usually laid only one egg at a time. It seems possible that when Captain James Cook first visited New Zealand in 1769, moa may have still survived in the remote areas in the western part of New Zealand's South Island. If so, these individuals would have been the last of their kind. Climatic conditions in New Zealand appear to have been relatively stable over the period during which moa became extinct. Different factors could have worked in concert to account for their abrupt disappearance. Vegetation was considerably altered by the Maori occupation of New Zealand, a change not easily explained by climate variation or other possible factors. Forest and shrubland burning appears to have reduced the prime habitat of many moa species. However, the main forest burning started around 700 years ago, after what current archaeological evidence indicates was the most intensive stage of moa hunting. While there appears to have been extensive burning on the east side of New Zealand's South Island, large forest tracts remained in the most southern part of the island. Because major habitat destruction seems to have occurred after moa populations already were depleted, and because some habitat that could have sheltered moa populations remained, it would seem that other factors were also at work in the extinction of these birds. For South Island, human predation appears to have been a significant factor in the depletion of the population of moa. At one excavated Maori site, moa remains filled 6 railway cars. The density of Maori settlements and artifacts increased substantially at the time of the most intensive moa hunting. This period was followed by a time of decline in the Maori population and a societal transition to smaller, less numerous settlements. The apparent decline fits the pattern expected as a consequence of the Maori's overexploitation of moa. Finally, the Maori introduced the Polynesian rat and the dog to New Zealand.
The actions of these potential nest predators could have reduced moa populations without leaving much direct evidence.
The Maori may have also inadvertently brought pests and disease organisms in fowls, which could have crossed over to eradicate moa populations. The possibility of analyzing ancient DNA to identify past diseases of extinct animals is being explored. However, evidence of such diseases is difficult to determine directly from paleoecological or archaeological remains. For these reasons, it is hard to determine the likelihood that introduced disease organisms were a cause of the decline of moa, but they are potentially significant. While the last of these possible causes remains speculative, defined clues exist for the action of the first two causes. The story of moa species and their demise raises ecological issues on the vulnerability of species to human-caused changes, including altered vegetative cover of the landscape, change in the physical environment, and modification of the flora and fauna of a region by eliminating some species and introducing others. 39-3 Forest fire suppression. Forest fires have recently increased in intensity and extent in some forest types throughout the western United States. This recent increase in fires has resulted partly from climate change and partly from human activities, for complicated reasons that foresters came increasingly to understand about 30 years ago but whose relative importance is still debated. One factor is the direct effect of logging, which often turns a forest into something approximating a huge pile of kindling. The ground in a logged forest may remain covered with branches and treetops, left behind when the valuable trunks are carted away. A dense growth of new vegetation springs up, further increasing the forest's fuel loads, and the trees logged and removed are of course the biggest and most fire-resistant individuals, leaving behind smaller and more flammable trees. Another factor is that the United States Forest Service in the first decade of the 1900s adopted the policy of fire suppression, for the obvious reason that it did not want valuable timber to go up in smoke or people's homes and lives to be threatened. The Forest Service's announced goal became put out every forest fire by 10 a.m. on the morning after the day when it is first reported. Firefighters became much more successful at achieving that goal after 1945, thanks to improved firefighting technology. For a few decades, the amount of land burnt annually decreased by 80%. That happy situation began to change in the 1980s, due to the increasing frequency of large forest fires that were essentially impossible to extinguish unless rain and low winds combined to help. People began to realize that the United States federal government's fire suppression policy was contributing to those big fires and that natural fires caused by lightning had previously played an important role in maintaining forest structure. The natural role of fire varies with altitude, tree species, and forest type. To make Montana's low-altitude ponderosa pine forest as an example, historical records, plus counts of annual tree rings and datable fire scars on tree stumps, demonstrated that a ponderosa pine forest experiences a lightning-lit fire about once a decade under natural conditions. The mature ponderosa trees have bark 2 inches thick and are relatively resistant to fire, which instead burns out the understory, the lower layer of fire-sensitive Douglas fir seedlings that have grown up since the previous fire. But after only a decade's growth until the next fire, those young seedling plants are still too low for fire to spread from them into the crowns of the ponderosa pine trees. Hence the fire remains confined to ground and understory. As a result, many natural ponderosa pine forests have a park-like appearance, with low fuel loads, big trees spaced apart, and a relatively clear understory. However, loggers concentrated on removing those big, old, valuable, fire-resistant ponderosa pines, while fire suppression for decades let the understory fill up with Douglas fir saplings that would in turn become valuable when full-grown. Tree densities increased from 30 to 200 trees per acre, the forest's fuel load increased by a factor of 6, and the government repeatedly failed to appropriate money to thin out the saplings. When a fire finally does start in a sapling-choked forest, whether due to lightning or human carelessness or intentional arson, the dense, tall saplings may become a ladder that allows the fire to jump into the crowns of the trees. The outcome is sometimes an unstoppable inferno. Foresters now identify the biggest problem in managing western forests as what to do with those increased fuel loads that built up during the previous half-century of effective fire suppression. In the wetter eastern United States, dead trees rot away more quickly than in the drier west, where more dead trees persist like giant matchsticks. In an ideal world, the Forest Service would manage and restore the forests, thin them out, and remove the dense understory by cutting or by controlled small fires. But no politician or voter wants to spend what it would cost to do that. 38:1 microscopes. Before microscopes were first used in the 17th century, no one knew that living organisms were composed of cells. The first microscopes were light microscopes, which work by passing visible light through a specimen. Glass lenses in the microscope bend the light to magnify the image of the specimen and project the image into the viewer's eye or onto photographic film. Light microscopes can magnify objects up to 1,000 times without causing blurriness. Magnification, the increase in the apparent size of an object, is one important factor in microscopy. Also important is resolving power, a measure of the clarity of an image. Resolving power is the ability of an optical instrument to show two objects as separate. 4. Example: what looks to the unaided eye like a single star in the sky may be resolved as two stars with the help of a telescope. Any optical device is limited by its resolving power. The light microscope cannot resolve detail finer than 0.2 micrometers, about the size of the smallest bacterium. Consequently, no matter how many times its image of such a bacterium is magnified, the light microscope cannot show the details of the cell's internal structure. From the year 1665, when English microscopist Robert Hooke discovered cells, until the middle of the 20th century, biologists had only light microscopes for viewing cells. But they discovered a great deal, including the cells composing animal and plant tissues, microscopic organisms, and some of the structures within cells. By the mid-1800s, these discoveries led to the cell theory, which states that all living things are composed of cells and that all cells come from other cells. Our knowledge of cell structure took a giant leap forward as biologists began using the electron microscope in the 1950s. Instead of light, the electron microscope uses a beam of electrons and has a much higher resolving power than the light microscope. In fact, the most powerful modern electron microscopes can distinguish objects as small as 0.2 nanometers, a thousandfold. Improvement over the light microscope. The period at the end of this sentence is about 1,000,000 times bigger than an object 0.2 nanometers in diameter, which is the size of a large atom. Only under special conditions can electron microscopes detect individual atoms. However, cells, cellular organelles, and even molecules like DNA and protein are much larger than single atoms. Biologists use the scanning electron microscope to study the detailed architecture of cell surfaces. It uses an electron beam to scan the surface of a cell or group of cells that have been coated with metal. The metal stops the beam from going through the cells. When the metal is hit by the beam, it emits electrons. The electrons are focused to form an image of the outside of the cells. The scanning electron microscope produces images that look three-dimensional. The transmission electron microscope, on the other hand, is used to study the details of internal cell structure. Specimens are cut into extremely thin sections, and the transmission electron microscope aims an electron beam through a section, just as a light microscope aims a beam of light through a specimen. However, instead of lenses made of glass, the transmission electron microscope uses electromagnets as lenses, as do all electron microscopes. The electromagnets bend the electron beam to magnify and focus an image onto a viewing screen or photographic film. Electron microscopes have truly revolutionized the study of cells and cell organelles. Nonetheless, they have not replaced the light microscope. One problem with electron microscopes is that they cannot be used to study living specimens because the specimen must be held in a vacuum chamber, that is, all the air and liquid must be removed. For a biologist studying a living process, such as the whirling movement of a bacterium, a light microscope equipped with a video camera might be better than either a scanning electron microscope or a transmission electron microscope. Thus, the light microscope remains a useful tool, especially for studying living cells. The size of a cell often determines the type of microscope a biologist uses to study it. 38:2 The raccoon's success. Raccoons have a vast transcontinental distribution, occurring throughout most of North America and Central America. They are found from southern Canada all the way to Panama, as well as on islands near coastal areas. They occur in each of the 49 states of the continental United States, although raccoons are native only to the Western Hemisphere. They have been successfully transplanted to other parts of the globe. Following a decline to a relatively low population level in the 1930s, raccoons began to prosper following their 1943 breeding season. A rapid population surge continued throughout the 1940s, and high numbers have been sustained ever since. By the late 1980s, the number of raccoons in North America was estimated to be at least 15 to 20 times the number that existed during the 1930s. By now, their numbers have undoubtedly grown even more, as they have continued to expand into new habitats where they were once either rare or absent, such as sandy prairies, deserts, coastal marshes, and mountains. Their spread throughout the Rocky Mountain West is indicative of the fast pace at which they can exploit new environments. Despite significant numbers being harvested and having suffered occasional declines, typically because of disease, the raccoon has consistently maintained high population levels.
Several factors explain the raccoon's dramatic increase in abundance and distribution.
First, their success has been partially attributed to the growth of cities, as they often thrive in suburban and even urban settings. Furthermore, they have been deliberately introduced throughout the continent. Within the United States, they are commonly taken from one area to another, both legally and illegally, to restock hunting areas and, presumably, because people simply want them to be part of their local fauna. Their appearance and subsequent flourishing in Utah's Great Salt Lake Valley within the last 40 years appears to be from such an introduction. As an example of the ease with which transplanted individuals can succeed, raccoons from Indiana have reportedly been able to flourish on islands off the coast of Alaska. The raccoon's expansion in various areas may also be due to the spread of agriculture. Raccoons have been able to exploit crops, especially corn, but also cereal grains, which have become dependable food sources for them.
The expansion of agriculture, however, does not necessarily lead to rapid increases in their abundance.
Farming in Kansas and eastern Colorado proceeded rapidly in the 1870s and 1880s, but this was about 50 years before raccoons started to spread out from their major habitat, the wooded river bottomlands. They have also expanded into many areas lacking any agriculture other than grazing and into places without forests or permanent streams. Prior to European settling and farming the Great Plains region, raccoons probably were just found along its rivers and streams and in the wooded areas of its southeastern section. With the possible exception of the southern part of the province of Manitoba, their absence was notable throughout Canada. They first became more widely distributed in the southern part of Manitoba, and by the 1940s were abundant throughout its southeastern portion. In the 1950s their population swelled in Canada. The control of coyotes in the prairie region in the 1950s may have been a factor in raccoon expansion. If their numbers are sufficient, coyotes might be able to suppress raccoon populations. By the 1960s, the raccoon had become a major predator of the canvasback ducks nesting in southwestern Manitoba. The extermination of the wolf from most of the contiguous United States may have been a critical factor in the raccoon's expansion and numerical increase. In the 18th century, when the wolf's range included almost all of North America, raccoons apparently were abundant only in the deciduous forests of the East, Gulf Coast, and Great Lakes regions, though they also extended into the wooded bottomlands of the Midwest's major rivers. In such areas, there are boreal habits and the presence of hollow den trees should have offered some protection from wolves and other large predators. Even though raccoons may not have been a significant part of their diet, wolves surely would have tried to prey on those exposed in relatively treeless areas. 38 to 3 transgenic plants. Genes from virtually any organism, from viruses to humans, can now be inserted into plants, creating what are known as transgenic plants. Now used in agriculture, there are approximately 109 million acres of transgenic crops grown worldwide, 68% of which are in the United States. The most common transgenic crops are soybeans, corn, cotton, and canola. Most often, these plants either contain a gene making them resistant to the herbicide glyphosate or they contain an insect-resistant gene that produces a protein called Bt toxin. On the positive side, proponents of transgenic crops argue that these crops are environmentally friendly because they allow farmers to use fewer and less noxious chemicals for crop production. For example,
A 21% reduction in the use of insecticide has been reported on Bt cotton.
In addition, when glyphosate is used to control weeds, other more persistent herbicides do not need to be applied. On the negative side, opponents of transgenic crops suggest that there are many questions that need to be answered before transgenic crops are grown on a large scale. One question deals with the effects that Bt plants have on non-target organisms such as beneficial insects, worms, and birds that consume the genetically engineered crop.
For example, monarch caterpillars feeding on milkweed plants near Bt cornfields will eat some corn pollen that has fallen on the milkweed leaves.
Laboratory studies indicate that caterpillars can die from eating Bt pollen.
However, field tests indicate that Bt corn is not likely to harm monarchs.
Furthermore, the application of pesticides has been demonstrated to cause widespread harm to non-target insects.
Another unanswered question is whether herbicide-resistant genes will move into the populations of weeds.
Crop plants are sometimes grown in areas where weedy relatives also live.
If the crop plants hybridize and reproduce with weedy relatives, then this herbicide-resistant gene will be perpetuated in the offspring.
In this way, the resistant gene can make its way into the weed
population.
If this happens, a farmer can no longer use glyphosate, for example, to kill those weeds. This scenario is not likely to occur in many instances because there are no weedy relatives growing
near the crop plant.
However, in some cases, it may become a serious problem.
For example, canola readily hybridizes with mustard weed species and could transfer its herbicide-resistant genes to those weeds.
We know that evolution will occur when transgenic plants grown on a large scale over a period of time.
Of special concern is the development of insect populations resistant to the Bt toxin.
This pesticide has been applied to plants for decades without the development of insect-resistant populations. However, transgenic Bt plants express the toxin in all tissues throughout growing season. Therefore, all insects carrying genes that make them susceptible to the toxin will die. That leaves only the genetically resistant insects alive to perpetuate the population. When these resistant insects mate, they will produce a high proportion of offspring capable of surviving in the presence of the Bt toxin. Farmers are attempting to slow the development of insect resistance in Bt crops by, for example, planting non-transgenic border rows to provide a refuge for susceptible insects.
These insects may allow Bt susceptibility to remain in the population. Perhaps the most serious concern about the transgenic crop plants currently in use is that they encourage farmers to move
farther away from sustainable agricultural farming practices, meaning ones that allow natural resources to continually regenerate over the long run. Transgenics, at least superficially, simplify farming by reducing the choices made by the manager. Planting a glyphosate-resistant crop commits a farmer to using that herbicide for the reason, probably to the exclusion of all other herbicides and other weed control practices.
Farmers who use Bt transgenics may not feel that they need to follow through with integrated pest management practices that use beneficial insects and timely applications of pesticides to control insect pests.
A more sustainable approach would be to plant non-transgenic corn, monitor the fields throughout the growing season, and then apply a pesticide only if and when needed. 37-2 Direct species translocation. It is becoming increasingly common for conservationists to move individual animals or entire species from one site to another. This may be either to establish a new population where a population of conspecifics has become extinct or to add individuals to an existing population.
The former is termed reintroduction and the latter reinforcement.
In both cases, wild individuals are captured in one location and translocated directly to another. Direct translocation has been used a wide range of plants and animals and was carried out to maintain populations as a source of food long before conservation was a familiar term. The number of translocations carried out under the banner of conservation has increased rapidly, and this has led to criticism of the technique because of the lack of evaluation of its efficacy and because of its potential disadvantages. The nature of translocation ranges from highly organized and researched national or international programs to ad hoc releases of rescued animals by well-intentioned animal lovers. In a fragmented landscape where many populations and habitats are isolated from others, translocations can play an effective role in conservation strategies, they can increase the number of existing populations or increase the size, genetic diversity, and demographic balance of a small population, consequently increasing its chances of survival. Translocation clearly has a role in the recovery of species that have substantially declined and is the most likely method by which many sedentary species can recover all or part of their former range.
However, against this is the potential for reinforcement.
Translocations to spread disease from one population to another or to introduce deleterious or maladaptive genes to a population.
Additionally, translocation of predators or competitors may have negative impacts on other species, resulting in an overall loss of diversity.
Last but not least of these considerations is the effort and resources required in this type of action, which need to be justified by evidence of the likely benefits.
Despite the large number of translocations that have taken place, there is surprisingly little evidence of the efficacy of such actions. This is partly because many translocations have not been strictly for conservation, neither have they been official nor legal, let alone scientific in their approach. Successful translocations inevitably get recorded and gain attention, whereas failures may never be recorded at all.
This makes appraisal of the method very difficult.
One key problem is a definition of success. Is translocation successful if the individuals survive the first week or a year, or do they need to reproduce for one or several generations? Whatever the answer, it is clear that a general framework is required to ensure that any translocation is justified, has a realistic chance of success, and will be properly monitored and evaluated for the benefit of future efforts. An example of apparent translocation success involves the threatened Seychelles warbler.
This species was once confined to Cousin Island, one of the Seychelles Islands, and reduced to 26 individuals. Careful habitat management increased this number to over 300 birds, but the single population remained vulnerable to local catastrophic events. The decision was taken to translocate individuals to 2 nearby islands to reduce this risk. The translocations took place in 1988 and 1990, and both have resulted in healthy breeding populations.
A successful translocation exercise also appears to have been achieved with red howler monkeys in French Guiana. A howler population was translocated from a site due to be flooded for hydroelectric power generation. The release site was an area where local hunting had reduced the density of the resident howler population. Released troops of monkeys were kept under visual observation and followed by radio tracking of 16 females. Although the troops appeared to undergo initial problems, causing them to split up, all the tracked females settled into normal behavioral patterns. Unfortunately, the success stories are at least matched by accounts of failure. Reviewing translocation of amphibians and reptiles, researchers C. Kenneth Dodd and Richard A. Siegel concluded that most projects have not demonstrated success as conservation techniques and should not be advocated as though they were acceptable management and mitigation practices. 36:1 Soil formation.
Living organisms play an essential role in soil formation. The numerous plants and animals living in the soil release minerals from the parent material from which soil is formed, supply organic matter, aid in the translocation and aeration of the soil, and help protect the soil from erosion. The types of organisms growing or living in the soil greatly influence the soil's physical and chemical characteristics. In fact, for mature soils in many parts of the world, the predominant type of natural vegetation is considered the most important direct influence on soil characteristics. For this reason, a soil scientist can tell a great deal about the attributes of the soil in any given area simply from knowing what kind of flora the soil supports. Thus, prairies and tundra regions, which have characteristic vegetations, also have characteristic soils.
The quantity and total weight of soil flora generally exceed that of soil fauna.
By far the most numerous and smallest of the plants living in soil are bacteria. Under favorable conditions, 1 million or more of these tiny, single-celled plants can inhabit each cubic centimeter of soil. It is the bacteria, more than any other organisms, that enable rock or other parent material to undergo the gradual transformation to soil. Some bacteria produce organic acids that directly attack parent material, breaking it down and releasing plant nutrients.
Others decompose organic litter to form humus.
A third group of bacteria inhabits the root systems of plants called legumes. These include many important agricultural crops, such as alfalfa, clover, soybeans, peas, and peanuts. The bacteria that legumes host within their root nodules change nitrogen gas from the atmosphere into nitrogen compounds that plants are able to metabolize, a process, known as nitrogen fixation, that makes the soil more fertile. Other microscopic plants also are important in soil development. For example, in highly acidic soils where few bacteria can survive, fungi frequently become the chief decomposers of organic matter. More complex forms of vegetation play several vital roles with respect to the soil. Tress, grass, and other large plants supply the bulk of the soil's humus. The minerals released as these plants decompose on the surface constitute an important nutrient source for succeeding generations of plants as well as for other soil organisms. In addition, trees can extend their roots deep within the soil and bring up nutrients from far below the surface. These nutrients eventually enrich the surface soil when the tree drops its leaves or when it dies and decomposes. Finally, trees perform the vital function of slowing water runoff and holding the soil in place with their root systems, thus combating erosion. The increased erosion that often accompanies agricultural use of sloping land is principally caused by the removal of its protective cover of natural vegetation. Animals also influence soil composition. The faunal counterparts of bacteria are protozoa. These single-celled organisms are the most numerous representatives of the animal kingdom and, like bacteria, 1 million or more can sometimes inhabit each cubic centimeter of soil. Protozoa feed on organic matter and hasten its decomposition. Among other soil-dwelling animals, the earthworm is probably the most important. Under exceptionally favorable conditions, up to 1 million earthworms may inhabit an acre of soil. Earthworms ingest large quantities of soil, chemically alter it, and excrete it as organic matter called casts. The casts form a high-quality natural fertilizer. In addition, earthworms mix of soil both vertically and horizontally, improving aeration and drainage. Insects such as ants and termites also can be exceedingly numerous under favorable climatic and soil conditions. In addition, mammals such as moles, field mice, gophers, and prairie dogs sometimes are present in sufficient numbers to have significant impact on the soil. These animals primarily work the soil mechanically. As a result, the soil is aerated, broken up, fertilized, and brought to the surface, hastening soil development. 36-2 Early ideas about deep-sea biology. In 1841, Edward Forbes was offered the chance to serve as naturalist aboard HMS Beacon, an English Royal Navy ship assigned to survey the Aegean Sea. For a year and a half the Beacon crisscrossed the Aegean waters. During that time Forbes was able to drag this small, triangular dredge, a tool with a leather net for capturing creatures along the sea bottom at 100 locations, at depths ranging from 6 to 1,380 feet. He collected hundreds of different species of animals, and he saw that they were distributed in 8 different depth zones, each containing its own distinct assemblage of animal life. The way zones of elevation on the side of a mountain are populated by distinct sets of plants. Forbes also thought he saw, as he later told the British Association, that the number of species and individuals diminishes as we descend, pointing to a zero in the distribution of animal life as yet unvisited. This zero, Forbes casually speculated, simply extended a line on his graph of animal number versus depth, probably began at a depth of 1,800 feet. Below that was the final zone in Forbes' scheme, Zone 9, a zone that covered most of the ocean floor and thus most of the solid surface of Earth. Forbes called this the Azoic zone, where no animal, to say nothing of plants, could survive. Forbes' Azoic zone was entirely plausible at the time, and it was certainly far from the strangest idea that was then entertained about the deep sea. In the first decade of the 19th century, a French naturalist named François Péron had sailed around the world measuring the temperature of the ocean. He found that the deeper the water, the colder it got, and he concluded that the seafloor was covered with a thick layer of ice. Péron ignored the fact that water expands when it freezes and that ice therefore floats. A more popular belief at the time was that water at great depth would be compressed to such a density that nothing could sink through it. This ignored the fact that water is all but incompressible. But even the more sensible naturalists of the day were guilty of a similar misconception. They imagined the deep sea as being filled with an unmoving and undisturbable pool of cold, dense water.
In reality, the deep is always being refreshed by cold water sinking from above. The central implication of all these misconceptions was that nothing could live in the abyss, just as Forbes's observations seemed to indicate. But Forbes erred in two ways. One was the particular study site he happened to use as a springboard for his sweeping postulate of a lifeless abyss. Although the Aegean had been the birthplace of marine biology, its depths are now known to be exceptionally lacking in animal diversity. Moreover, through no fault of his own, Forbes was not particularly successful at sampling such life as did exist at the bottom of the Aegean. It was his dredge that was inadequate. Its opening was so small and the holes in the net so large that the dredge inevitably missed animals. Many of those it did catch must have poured out of its open mouth when Forbes reeled it in. His Azoic zone, then, was a plausible but wild extrapolation from pioneering but feeble data. As it turned out, the existence of the Azoic zone had been disproved even before Forbes suggested it, and the theory continued to be contradicted regularly throughout its long and influential life. Searching for the Northwest Passage from the Atlantic to the Pacific in 1818, Sir John Ross had lowered his deep-sea clam, a sort of bivalve sediment scoop, into the water of Baffin Bay, which they determined to be more than 1,000 fathoms deep in some places. Modern soundings indicate he overestimated his depths by several hundred fathoms, but in any case Ross's clam dove several times deeper than Forbes's dredge. It brought back mud laced with worms, and starfish that had entangled themselves in the line at depths well below the supposed boundary of the Azoic zone. 35-3 Seasonal succession in phytoplankton. Phytoplankton are minute, free-floating aquatic plants. In addition to the marked changes in abundance observed in phytoplankton over the course of a year, there is also a marked change in species composition. This change in the dominant species from season to season is called seasonal succession, and it occurs in a wide variety of locations. Under seasonal succession, one or more species dominate the phytoplankton for a shorter or longer period of time and then are replaced by another set of species. This pattern is repeated yearly. This succession is different from typical terrestrial ecological succession in which various plants replace one another until finally a so-called climax community develops, which persists for many years. What are the factors causing this phenomenon? Considering that seasonal succession is most often and clearly seen in temperate seas, which have a marked change in temperature during a year, temperature has been suggested as a cause. This may be one of the factors, but it is unlikely to be the sole cause because there are species that become dominant species at various temperatures. Furthermore, temperature changes rather slowly in seawater, and the replacement of dominant species often is much more rapid. Another suggested reason is the change in nutrient level over the year, with differing concentrations favoring different phytoplankton species. While this factor may also contribute, observations suggest that phytoplankton populations rise and fall much more quickly than nutrient concentrations change. Yet another explanation is that species succession is a consequence of changes in seawater brought about by the phytoplankton living in it. Each species of phytoplankton secretes or excretes organic molecules into the seawater. These metabolites can have an effect on the organisms living in the seawater, either inhibiting or promoting their growth. For any individual organism, the amount of metabolite secreted is small. But the effect of secretions by all the individuals of the dominant species can be significant both for themselves and for other species. These organic metabolites could, and probably do, include a number of different classes of organic compounds. Some are likely toxins, such as those released by the dinoflagellates during red tides, which inhibit growth of other photosynthetic organisms. In such cases, the population explosion of dinoflagellates is so great that the water becomes brownish-red in color from the billions of dinoflagellate cells. Although each cell secretes a minute amount of toxin, the massive dinoflagellate numbers cause the toxin to reach concentrations that kill many creatures. This toxin can be concentrated in such filter-feeding organisms as clams and mussels, rendering them toxic to humans. Another class of metabolite is the vitamins. It is now known that certain phytoplankton species have requirements for certain vitamins, and that there are considerable differences among species as to requirements.
The B vitamins, especially vitamin B12, thiamine and biotin, seem to be the most generally required. Some species may be unable to thrive until a particular vitamin, or group of vitamins, is present in the water. These vitamins are produced only by another species, hence, a succession of species could occur whereby first the vitamin-producing species is present and then the vitamin-requiring species follows. Other organic compounds that may inhibit or promote various species include amino acids, carbohydrates, and fatty acids. Although it is suspected that these organic metabolites may have an important role in species succession, and it has been demonstrated in the laboratory that phytoplankton species vary both in their ability to produce necessary vitamins and in their requirements for such in order to grow, evidence is still inadequate as to their real role in the sea. There is also evidence to suggest that grazers, particularly selective grazers, can influence the phytoplankton species composition. Many copepods and invertebrate larvae pick out selected phytoplankton species from mixed groups, changing the species composition. A growing body of evidence now suggests that all of the factors considered here are operating simultaneously to produce species succession. The importance of any factor will vary with the particular phytoplankton species and the environmental conditions. 54 to 3 elements of life. The creation of life requires a set of chemical elements for making the components of cells. Life on Earth uses about 25 of the 92 naturally occurring chemical elements, although just 4 of these elements, oxygen, carbon, hydrogen, and nitrogen, make up about 96% of the mass of living organisms. Thus, a first requirement for life might be the presence of most or all of the elements used by life. Interestingly, this requirement can probably be met by almost any world. Scientists have determined that all chemical elements in the universe besides hydrogen and helium were produced by stars. These are known as heavy elements because they are heavier than hydrogen and helium. Although all of these heavy elements are quite rare compared to hydrogen and helium, they are found just about everywhere. Heavy elements are continually being manufactured by stars and released into space by stellar deaths, so their amount compared to hydrogen and helium gradually rises with time. Heavy elements make up about 2% of the chemical content of our solar system, the other 98% is hydrogen and helium. In some very old star systems, which formed before many heavy elements were produced, the heavy element share may be less than 0.1%. Nevertheless, every star system studied has at least some amount of all the elements used by life. Moreover, when planetesimals, small, solid objects formed in the early solar system that may accumulate to become planets, condense within a forming star system, they are inevitably made from heavy elements because the more common hydrogen and helium remain gaseous. Thus, planetesimals everywhere should contain the elements needed for life, which means that objects built from planetesimals, planets, moons, asteroids, and comets also contain these elements. The nature of solar system formation explains why Earth contains all the elements needed for life, and it is why we expect these elements to be present on other worlds throughout our solar system, galaxy, and universe. Note that this argument does not change, even if we allow for life very different from life on Earth. Life on Earth is carbon-based, and most biologists believe that life elsewhere is likely to be carbon-based as well. However, we cannot absolutely rule out the possibility of life with another chemical basis, such as silicon or nitrogen. The set of elements used by life based on some other element might be somewhat different from that used by carbon-based life on Earth. But the elements are still products of stars and would still be present in planetesimals everywhere. No matter what kinds of life we are looking for, we are likely to find the necessary elements on almost every planet, moon, asteroid, and comet in the universe. A somewhat stricter requirement is the presence of these elements in molecules that can be used as ready-made building blocks for life, just as early Earth probably had an organic soup of amino acids and other complex molecules. Earth's organic molecules likely came from some combination of 3 sources: chemical reactions in the atmosphere, chemical reactions near deep-sea vents in the oceans, and molecules carried to Earth by asteroids and comets. The first 2 sources can occur only on worlds with atmospheres or oceans, respectively. But the third source should have brought similar molecules to nearly all worlds in our solar system.
Studies of meteorites and comets suggest that organic molecules are widespread among both asteroids and comets.
Because each body in the solar system was repeatedly struck by asteroids and comets during the period known as the heavy bombardment, each body should have received at least some organic molecules.
However, these molecules tend to be destroyed by solar radiation on surfaces unprotected by atmospheres.
Moreover, while these molecules might stay intact beneath the surface, they probably cannot react with each other unless some kind of liquid or gas is available to move them about. Thus, if we limit our search to worlds on which organic molecules are likely to be involved in chemical reactions, we can probably rule out any world that lacks both an atmosphere and a surface or subsurface liquid medium, such as water. 51-2 Surface fluids on Venus and Earth. A fluid is a substance, such as a liquid or gas, in which the component particles can move